Good morning. My name is Ranmal Samarasinghe, and today I'm going to be talking about human brain organoids as a stem cell-based research tool for neuroscience. So, let me start this. I am an MD PhD neurologist and a new assistant professor in the Department of Neurology at UCLA, and I'm also a clinician scientist, and so I see patients with epilepsy and autism. The Broad Stem Cell Research Center has been really important in my research career. I did all the work that you're going to see today in Dr. Ben Novich's lab, and he's a center researcher. He's my postdoctoral mentor. We've had a close collaboration with Bill, who's speaking today. And I'm the recipient of Broad Stem Cell Research clinical fellowship that allowed me to actually complete my postdoctoral work and has resulted in me starting my own lab recently, where I'm interested in using stem cell technology to understand brain disease, in particular epilepsy and autism, but hopefully with implications for other neurological disorders. So how do we hope to do this? So modeling the brain and physiology is complicated. And the point of this first image is just to show that the human brain is large, it's complex, it's got a lot of parts, and it's bigger than a lot of the models that we use to study brain in the laboratory, like mouse brain or monkey brain. The human brain is bigger, that is. The brain is also this physiologically active organ. And so what you're seeing here are actually brain waves from a patient, from something called an EEG, or an electroencephalogram. And EEG is obtained in this way, where we put it's a cartoon showing electrodes placed on the scalp of a patient, and we can get this kind of a signal. And clinically, as an epileptologist and a clinical physiologist, this is something that we do routinely, and it provides information on brain health. But at a deeper level, what the EEG actually shows is a coordinated activity of brain cells, of neurons within the brain, that are firing in such a way as to be able to produce these brain waves. So the task at hand is to go from a stem cell and produce this model that that is able to reproduce this anatomical complexity that you see in human brain, as well as this brainwave complexity, right? This electrical complexity. So how could we possibly do this? So our approach is to really go from this really big, complicated organ, complicated problem, and try and simplify it. And so I'm showing illustrations here of a brain and of like a picture of a circuit, of a computer circuit, essentially, that looks complicated and has a lot of parts. And the idea is to take a small piece of this, of each of these, as an example, and then extrapolate from that small piece into the larger problem. And that really is fundamentally how I think of you going from stem cells and using brain organoids as a tool. So what is a brain organoid? So it's derived from a stem cell, including from a human stem cell of a patient that has a particular disease. And this is an image of a brain organoid. It is this three-dimensional structure that's grown in a laboratory dish, and that's brain-like. So why is it brain-like? Because in this cartoon, what you're seeing is it has components that are seen in actual human brain, components that are actually not seen, sometimes seen in other brain, like rodent brain. And it has this really nice anatomical organization. This is a microscope image of an organoid, and the different colors represent different cells that are separated, just like you would see an actual brain. Now, we knew all this when I started my research in Ben's lab in the BSCRC, but what we were more interested in knowing is, does this also result in sophisticated EEG-like activity in brain waves? And so to do this, of course, we have to come up with some new tools to use with organoids. And we have two major tools that we use. And one is using a sophisticated microscope called a multiphoton microscope to look at the actual activity of individual neurons within an organoid. So what you're seeing here is a brain organoid derived from a stem cell. And the flashes you're seeing is individual cells lighting up during a recording. And then we've developed tools to actually look at that data and pick up the individual cell firing patterns. The other tool that we use, other than this multiphoton calcium indicator imaging, is kind of like an EEG for an organoid. It's called a local field potential recording. What you're seeing here is a picture of the actual process. So we make these electrodes and we stick it directly into the organoid, and we can get an image, or excuse me, data like this. And so what you're seeing here, if you can see my cursor, are these peaks and valleys. And each of these peaks actually represent the actual brain waves from the organoid. And remarkably, these brainwaves actually look like what we'd see in an actual human patient. So we can reproduce some of that physiological complexity in human brain in the organoid. And so just to recap briefly, we can make organoids from a patient's own stem cells. It has a sophisticated anatomy. And what we've really been working on to push this technology even further is to measure brainwaves in the organoid. And we saw very early on that these brainwaves are actually similar in the organoids to what are seen in an actual human brain. Okay, so how are we applying this? So for our first studies, we actually had Bill's help because Bill had stem cells from a patient that has a disorder that's associated with autism and epilepsy. It's called Ret Syndrome. What you're seeing here is the calcium indicator imaging that I described earlier from a normal organoid. So this is a brain that doesn't have any disease. And you're seeing, hopefully in the video, individual cells firing one after the other. And below is a plot where every yellow kind of streak represents an individual cell. In contrast, when we made an organoid from a patient with the Rett syndrome, with this genetic disease, it looks different. And I hope you can appreciate this in the video, but what you're seeing actually is individual cells firing, as well as this kind of the entire organoid firing together in these flashes. And I've shown that below in the plot where we have the yellow streak kind of all going together. And that's what's highlighted in the red dotted boxes. And these flashes are actually like what are seen sometimes in patient EEG. It's very similar to an epileptic-like state. And in other words, we can go from the patient stem cell to the organoid and recreate some of the pathology that we'd expect from physiological measurements in an actual patient. And of course, I said we're using two techniques. So what about the other technique I described, this organoid EEG approach? So here's a normal, but to describe the normal, let me show you the patient. So this is a Rett patient again in red. And at the top, what you're seeing is the raw trace, what we call when I first stick the electrode into the organoid, this is what I see. And the raw trace in the normal is this kind of smooth crescendo decrescendo figure, right, in blue. In the patient, it looks really different. We're seeing all these lines sticking out, and these are called spikes. It's highly abnormal. Then when we go to look at the actual EEG part of it, where we want to see these peaks and valleys, we see that in the normal. None of that in the case of the Rett syndrome organoid. It's just like a flat descending line. And so what we learned from this is that we can reproduce this epileptic-like state in the organoid from a patient that should have epilepsy. And we use this to learn more about why this is. And I won't get into that here in the interest of time. Instead, I'll talk about another important potential use of organoids and that we tested with Rett syndrome. And that's in application in drug discovery. So as I mentioned, Rett syndrome is associated with epilepsy. Epilepsy is a disease without a medical cure. The best you can do for a patient with epilepsy is give them drugs that they have to then take for the rest of their lives. That's the best you can do. And so epilepsy, by the way, is defined by recurrent spontaneous seizures. And unfortunately, for 1 out of 3 patients, about 30% of our patients don't even respond to the newest drugs out there. So despite the best drug treatment available, they still have seizures. And this hasn't improved in over 100 years of trying. And so what does this suggest to us? We need new models to try and understand epilepsy and to treat the disease and to come up with new drugs. And maybe organoids are one of these models. So here is the Rett EEG recording, EEG-like recording from a Rett organoid without any drugs. And so you see a lot of these spikes like that I showed before, and you don't see brain waves. Then we tried to add a traditional anti-seizure medication. In this particular case, we actually saw fewer spikes, which is good, but we didn't see any brain waves coming up. So that's not good. Then we tried a novel drug called pifithrine alpha. Once again, this is from Bill and his own studies in Rett, where he found that this drug can be effective. And we saw a reduction in spikes, just like with the traditional drug. But what was really great is we actually also saw some restoration of brain waves, suggesting that in this model, using this novel drug, we can get more of a rescue than we've seen before using other models and using other drugs that are traditionally used to treat Rett syndrome, at least epilepsy and Rett syndrome. So in conclusion, essentially what we were able to do is use the model that we have with organoids in this particular disease to, first of all, see abnormality, find out why that abnormality is happening, and to test some new drugs. So where are we going with this? One idea is personalized medicine. And so what do I mean by that? Well, this is an example of an EEG, and it's really abnormal. It's from a patient that has a really, really devastating genetic epilepsy that basically is present at birth. That around 4 months is when this happens, and it has no cure. Here is our calcium indicator recording from organoid with the same genetic mutation, right? So you can see the similarity, but these are different patients. The idea, sort of the goal in the long term, is to go from a single patient, get stem cells from that patient, make organoids from that patient, and then, for example, test drugs so that you can have personalized therapy for that patient, or maybe even personalized understanding of why that patient has that disease. So that's one sort of future direction. The other idea is I don't want these organoids just to be used for what I'm interested in, which are epilepsy and autism, for example, but broadly used to study brain disease. What I didn't mention earlier when I said we're making brain organoids, specifically, we can actually make brain organoids for very specific parts of the brain, including this part of the brain called the hippocampus. And the hippocampus is where human memories, or where memories generally for any animal, where our memories are formed. And so we did make hippocampal brain organoids, and what we discovered is that these hippocampal organoids have brain waves that are very similar to what is actually made in real hippocampus, in an animal or in a human. And these brain waves are associated with memory formation and memory consolidation. And so this is really remarkable, and as many of you know, there are a lot of diseases where memory is affected. And so we've been working with some of our collaborators that work in these disease areas, like Dr. Chow Peng in Parkinson's disease and Dr. Jess Rexek in Alzheimer's. And the goal now, for example, here is to make organoids that have pathology associated with memory loss. And then we can see what happens to these brain waves. Are they gone? Why are they gone if they aren't? And if they don't go, why is that? So really understand disease mechanism. So in summary, what I really wanted to illustrate overall in terms of brain organoids and their utility is the way that I think about an organoid. And that is as this tool to really unlock the awesome potential of a stem cell, to go from this simple yet very highly potentiated thing, the stem cell, to something that we can then use to study even more sophisticated problems. And really, in my mind, nothing gets more sophisticated than the brain. So we can use the organoid now to study brain anatomy and also brain physiology. And with that, hopefully, go forward in enhancing our understanding of neurological disease processes and come up with new therapies that we haven't in the past. So inclusion, thank you. This has been really collaborative work throughout UCLA, as well as from collaborators outside of UCLA. I've had a lot of funding sources that have been important. Certainly the Broad Stem Cell Research Center has been one of those. Thank you.